Okay, cool. All right, so for the second seminar of the semester, we have Casey coming from the Minnesota DNR. Casey coordinates the efforts of the Sentinel Lakes long-term monitoring program, which is a collaborative effort between Minnesota Department of Natural Resources and the Minnesota Pollution Control Agency. Collaborators within the Sentinel Lakes program intensively monitor multiple trophic levels in 25 lakes throughout the state, representing a range of lake types and ecoregions to better understand how and why Minnesota lakes are impacted in response to stressors such as changing climate, landscape, and invasive species. And just as me using Sentinel Lakes data, Sentinel Lakes is a really awesome program, so we're really delighted to have Casey here speaking about some of the work from Sentinel Lakes. And with that, I will turn it over to Casey.
Perfect, thank you. Okay, thank you.
Yeah.
All right. So yeah, no, thank you for the invitation and the opportunity to share some of the things that we've been learning about. And before we begin, want to recognize some of the other members of the program that are here today as well. Will French and Tim Martin, and then myself. The three of us are the only the full-time members of the Sentinel Lake program, and I'll get into a little bit more later about all the other folks who are involved with those efforts. But yeah, this is exciting to present and share some of these data. This is the first group to see a lot of the data that are going to be presented. So this is very active exploration right now, and so curious to get feedback and thoughts and kind of as we go. And so but yeah, the bulk of the presentation is going to focus on lake temperatures and where we're seeing changes and things like that, and exploring that data set. But I also want to introduce everybody to the program itself. Just a little bit of a brief introduction about what we do and who we are. The bulk of the presentation is going to focus on the water temperature data set, like I mentioned, because we're seeing some really interesting things. And so wanted to share that with this group. Then the last part of the presentation, want to take that back to more fisheries application, and so show you a couple of different ways that these changes are that we're seeing these changes manifest themselves in the fish community and different aspects of the fish community that we're looking at as well. And then of course finish up. Should have plenty of time for Q&A and hopefully a discussion. So just really broad, the program's focus are kind of these four pillars here. The largest, most important being our monitoring aspect of it. And that's through consistent, standardized, long-term monitoring. That's really our bread and butter to really understand what's going on in Minnesota's lakes. And through that, we're discovering all kinds of new things. That's why I'm here, going to share some of those highlights of things that we're finding, better understanding not only what changes are occurring, but the mechanisms behind those changes as well. And also, yeah, in part why we're here is to help inform the scientific community of what we're seeing. So we have responsibilities to inform not only DNR and PCA and things like that, where management decisions are in this application is being made, but to share it with students and faculty and the public at large as well. And then the fourth is supporting scientific research, and so like external research, like what occurs here at the University of Minnesota and other University of Minnesota or other institutions, both within Minnesota and outside of Minnesota. So this is an incredible framework that can be used for a lot of different ways. And so we're always happy to support those efforts where the data where it makes sense to do that. So the program started back in 2008, and like Chris mentioned, this is a collaborative effort with DNR and PCA. So there's a lot of, and I'll get into in a couple more slides here, all the different agencies that play a role in this effort. And so it's very complementary in that way. And we actually got our start through LCCMR, through several rounds of LCCMR funding, got this program up and running, and eventually, more recently, in the terms of the program, is that we've switched, our 3 positions are now permanently funded through DNR, and so which is I don't know, just as fantastic, I guess. So that we have that that solid footing moving forward. So, but a lot of the effort, and I'll go into that a little bit further, but the 3 of us, yeah, we play a role, but largely the efforts toward this program are through in-kind support, through biologists and programs that don't receive direct funding from the program. And that transitions into this next slide, because this really highlights the different trophic levels that we're monitoring, and the biologists, the agencies that play a role in collecting those data. And so, and that varies from pollution control agency. The general thought is to have the experts in that field, those topic level experts, play the lead role in collecting the data for that trophic level, right? PCA is they're the experts with water quality, and so they go out in the tier 1 lakes. PCA is on the water 1 month for 6 months of the open water season of 12 of the Sentinel lakes. And so there's a monthly record of water quality on the tier 1 Sentinel lakes because of their efforts. In addition to PCA, some of the things that DNR Fisheries and Sentinel Lakes program puts a lot of effort into, and we're going to talk a lot about it today, is our water temperature. Some of our continuous loggers looking at water temperature, dissolved oxygen, lake levels, and things like that. And so we'll talk about those things. The water temperature loggers are in all 25 of the Sentinel lakes to varying degrees. Whereas dissolved oxygen, this is continuous dissolved oxygen. When PCA goes out, they take discrete profiles, and when the fisheries areas go out for lake surveys, they take discrete profiles. So there are records of a lot of those data. But we're transitioning more toward, as monies become available, toward more continuous monitoring. So all 25 lakes have continuous water temperature monitoring, but only a subset have dissolved oxygen, continuous dissolved oxygen monitoring, in part because of the expense involved with that. And water level, and this has really become I don't know, the need for this has become more heightened lately as we enter different drought cycles and things like that. But we also maintain continuous lake level loggers on the tier 1 Sentinel lakes as well. And then citizen scientists play a large role in collecting lake level information from the tier 2s, just using a staff gauge. And so those data are also available. And then we work with the state climatology group for weather and ice on and ice off. They maintain the whole state has a really nice record of ice on, ice off data in Minnesota, and that's EWR is within DNR. Yeah, some of the other trophic levels. Phytoplankton is available on a subset of the Sentinel lakes. And we just recently moved to biovolume on the phytoplankton, taxa-specific biovolume on some of these lakes, which is really nice. Zooplankton, macroinvertebrates, aquatic plants. So EWR has a program dedicated with a number of different research scientists that have different trophic level expertise. There's several several of those scientists that specialize in aquatic plants, other ones that specialize in zooplankton, and other in macro invertebrates. And so this is back to like, this is all in-kind support again. And so this, all of this effort is, just because people believe in the efforts of the program and the strength of that collective data set, right? And leveraging all these things together. We're housed in the Fisheries Research Unit, and so we come at the program. My background's in fisheries. We come at this from more of a fish aspect, and that's where it's great to work with all these other scientists that are experts in these other trophic levels. So we work with the fisheries section who completes all the standard surveys in not just the Sentinel Lakes, but statewide, right? And those are standard gill nets and trap nets that you think of, focused on walleye and pike and things like that. What we do is we kind of fill in some of the gaps where those surveys maybe don't collect all of that information. And so we focus on a subset of lakes and sampling juvenile fish. So we go out in the fall and we'll electrofish for age 0 fish. So they were born that spring, and we sample them in the fall to see how big they've gotten, a variety of different ecologically, more sensitive species, largemouth bass, bluegill, yellow perch, and rock bass. And so we see how big they are. And some of those bluegill could be, 20, 30 millimeters by the fall of their first year. That has implications for going into that first winter, how probability of survival and things like that, So, and I'll get into how the water temperature plays a large role in that as well. Pelagic fish. So cold water pelagic fish. So Cisco, some lake trout and things like that, and primarily Cisco. We put a lot of, our program puts effort into targeting Cisco, directly targeting Cisco to monitor those populations. And at when we talk about the different applications at the end, we'll go into some of those data as well. Then there's also the Fish IBI program, and they go out and sample nearshore fish communities, again, that are not represented in the standard fish surveys. And so several aspects of the fish community together with these other trophic levels. And then data management. And so Tim is our data scientist. And so there's a lot of data that comes in from these different areas. And it's our responsibility to make sure that we're compiling that and cleaning it. And Tim's developed a lot of different QA/QC processes and really streamlined a lot of what we do in that regard to give us really good data sets. So I wanted to touch briefly on how these lakes were selected, because it's going to be important in our conversation for when we start talking about the water temperature data. So Minnesota is really interesting, right? In that it varies in landscape from the Southwest all the way up to the Northeast. And we wanted to represent all the different lake types that are possible along that spectrum. And so the lakes were strategically selected to occur within each of these 4 land types. I'm going to use the term ecoregion quite a bit, and that's kind of true. The southern 3 are ecoregions. We've also kind of separated out the Canadian Shield Lakes as their own group. They're not technically their own ecoregion, but they behave so differently. The lakes are so different that we're going to give them their own grouping here. And you'll see when we start looking at water temperature data, the fish data, I mean, you look at any of the biological components and the thermal components, and they're just different. Okay, so they get their own special category up there. Okay, well, we want to represent different lake types within each of these 4 land use types. Wanted both stratified and mixed lakes, and wanted those to occur along a spectrum of lake productivities. So within each of the ecoregions, there would be 3 stratified and 3 mixed sentinel lakes, each representing that, spectrum of productivity. So then we have that for each of the 4 ecoregions, giving us 24 total lakes. A 25th was added for good measure, Greenwood Lake, because it's a natural reproducing lake trout population. And I'm sure glad, and I know that Will and Tim are glad that we added Greenwood, because it's a gem of a lake up in the Northeast. So, but that's how we got the lake. So these lakes, they represent that spectrum. So within all the Minnesota lake types, there are some of the sentinel lakes represent a lot more. When we talk about representation, some of them represent a lot of lakes, and some of them don't represent very many lakes, but it gives you that, it's that spectrum, it's that variability that we're capturing. Again, so when we talk about water, we're going to talk about water temperature, it's that spectrum. It's not, these aren't averages or things like that. It's that range that we're focused on. So these are the 25 sentinel lakes, and across those, across the different, and it is amazing. We work, all the way from the Southwest Oshakatan it only gets about 11 feet deep. It's a really productive lake. It should belong in South Dakota, is where that lake actually should belong. All the way up to, like I mentioned, Greenwood, which is lake trout, Cisco, just a beautiful lake, clear water, and then everything in between. And then the central part of the state, just a lot of those typical Minnesota lakes, both shallow and deep, walleye lakes and bass lakes. There's a really good variety of lakes represented. Okay, so move on to the water temperature. So I wanted to start with a little bit of some previous work that's been done to set up the conversation a little bit for the water temperature data that we're going to show you. And so in 2015, this was a landmark paper by O'Reilly and a bunch of different co-authors, and they were able to quantify increases in water temperature in lakes around the world. So this is surface water temperature during the summer. So specific location of the lake, specific time of year, but the spectrum is just impressive. You'll notice that not all trends are consistent, right? And that's okay, but on average, there was an increase. So just being able to show that is a huge step forward. But it also begs the question of, okay, are changes occurring in other times of the year? Are they occurring at other depths of the lake? And so there's a lot of this was a huge leap forward in documenting these some of these changes. So speaking of the seasonal aspect, this Winslow paper was done using 5 LTER sites based in Trout Lake, which is like a northern Wisconsin long-term monitoring station. And what's neat about this paper is that they used discrete profiles. So discrete profiles are commonly available. And this, I forget the years of this particular paper, but what was unique about it, what it showed is that water temperature was changing differently depending on the month. So that's interesting. So they're taking it one step further. But it also kind of highlights the need for, so these previous papers, based on discrete profiles and things like that. And that's what we've had. But as technology continues to improve, we've moved more and more to continuous loggers and things like that. But what's kind of interesting is that okay, these loggers have been around for a while, but you actually need them in the lakes for periods of time to kind of gather these data sets, right? And to make sure that these data sets are talking to each other and that we're doing things in a standardized fashion across the landscape and things like that. So that's where our program comes in. Like I mentioned, so back in 2008, we started putting these temperature loggers in all 25 of the Sentinel lakes. There's currently over 180 loggers in the 25 lakes. That's because some of the lakes have chains of loggers like the one depicted here, so that we can look at things like duration of stratification and depth of stratification and things like that over time. But some shallow lakes have just a simple like one logger or maybe two loggers, things like that. So it really varies depending on the lake. We collect data at 1 hour intervals. You can set them up to record at whatever frequency you want. We kind of settled on an hour as kind of a happy in between of yeah, for our purposes, we don't need it too frequent. And the longer you go, like if you do about 1 hour, that gives you about a 5-year battery life on these units. And we've, like for our needs, this 1 hour has worked out actually pretty well. We place these in the deepest basin of the lake. So the data that we're going to show you is this pelagic data, but we're going to focus on the shallowest loggers of each of these lakes. So these are all epilimnion loggers, but they're out deep. We also, with the lake level monitoring, the continuous loggers, those also record water temperature. But we're not including that with this. We're just focusing on the deep water. So they're out in the middle of the lake, but it's the shallowest logger available. Okay, so some of the questions that we're going to focus on. I broke this into 2 questions, part 1, questions part 2 for water temperature. So to start off, we're going to start very big picture and are changes occurring to water temperature in Minnesota lakes? Okay, so we'll cross that first, and then we'll get into more specifics about are those changes uniform throughout that annual cycle? And that's something we were really interested in. Okay, think about whatever you studied. Okay, I'll take a fish's, a fish life history and think of all the things that occur throughout a course of a year. If you're interested in zooplankton, if you're interested in aquatic plants, whatever it is, think about how water temperature is really impactful for different processes. Or if you're into nutrient cycling, think of, with stratification and things like that. It's important for us to understand when water temperature is what it is, and are those temperatures changing over time. And the other thing is, because we're looking at the span of Minnesota, where are changes occurring? Okay, and so is it specific? Is it uniform across all these ecoregions, or is it specific to certain areas? So let's start there with part 1. So for part 1, we're going to use a couple of different metrics. And again, as a happy medium, we kind of settled on weekly means. Daily means were a little too specific, and monthly maybe a little too broad. So we settled on weekly, but there's really not a huge rhyme or reason for that. That just seemed to be a happy medium. So we're going to present the data as a weekly mean in one case. And then we'll also present it as a weekly mean for the cumulative growing degree days. And so doing this as more of a metric to capture or try to understand the impact to fish in particular, but other biological organisms as well. And so looking at it a little bit different way. So growing degree days with a base of 5. So that's just everything over 5 degrees Celsius, and summing that up just to get an understanding of those of those cumulative thermal units. Okay, so we'll start into the data, and I have a note here to remind myself to slow down and explain this figure and what's involved with it, because we're going to see a lot of similar figures as we go through. So on the Y, you have that mean temperature, and you can see kind of where it, if you let 20 degrees Celsius kind of be your guide, you can see where the different the lakes kind of first achieve that, and then pass that in the fall. The different colors represent the 4 different ecoregions, right? And so if you start at or if you're up in the summer, they're just inversely related to the latitude of that ecoregion, right? I mentioned those Canadian Shield Lakes are their own group. So if you just focus on summer temperatures, they're obviously a lot lower than the other ecoregions. The other symbols to keep in mind, we're going to use these consistently throughout, is that the closed circles represent increasing trends and the open squares represent decreasing trends. Okay. So let's see if I kind of highlight this, oh, it's not showing up. That's okay. This early springtime right here, early springtime, there's kind of a cluster of those open squares, right? So there might be trends of decreasing weekly water temperatures over time. But as you go up and you make your way into spring, there's a lot of closed circles representing a lot of increasing trends over time. And then as it kind of descends there on the back, there's kind of a mixture. You see some open squares that are red, meaning the glacial shield lakes, and you see some closed circles representing some increases there. But this figure includes all trends, regardless of whether they're meaningful or not. And so this is just to remind me to point out to you that not all these trends are meaningful, right? And so if we focus in on the statistically significant trends that we saw in weekly mean temperature, I have those summarized here. There were 35 of over 200 possibilities. So by week and by the 4 ecoregions, so of over 200 possibilities, 35 of those were significant. 30 of those were positive trends, 5 were negative trends. This little makeshift table just shows you when those weekly trends were occurring, whether they were positive or negative. And you can see that the bulk were positive in June and July. Okay. And if we look at that as a figure, so this is just weighting the color by the significance value, but it really does a nice job of showing those June and July trends in weekly temperature, right? And so this is, I really like this figure, and just really demonstrating kind of one of the take homes that we want you to learn is when those changes are occurring. And it just demonstrates that really well. Look at all those filled circles in that June, July. I should point out, so I need a ruler. But for those online, they can probably see my cursor. But so June is weeks like 23 to 26, and July is like 27 to 31. So that cluster is both June and July, and you can kind of see where July is because it's that summer stability with both July right before the peak. So now let's go to weekly growing degree days, and so this is a similar plot weighted by significance for growing degree days. And it really kind of highlights the same thing that we saw with those weekly trends, with that focus in June and July. If we look at the actual significant trends based on just based on the stats of it, there were fewer significant trends. And so I was able to just put them up here just to kind of illustrate a few general points. All of these trends were in the positive direction for growing degree days. All but one occurred during June. There was one during July. The rest were occurring during June. And none of the significant trends occurred in the glacial shield ecoregion, which is also really interesting. So positive during June, we're adding growing degree days in that month more than at least, yeah, we're adding growing degree days in June. And to look at the actual weeks is kind of interesting. So weeks, there's a lot of support for week 24 in this case, and then secondarily week 25. So we can look at those more specifically. Here's week 24 plotted by ecoregion. So this is the increasing trend in growing degree days for that particular week, right? So from 2008, so we have 15 years of trend data, and you can see how the southern three ecoregions are increasing at fairly similar rates, and then the Canadian Shield Lakes are doing their own thing. And then this is week 25. Again, you can see how the steepest slope is actually northern lakes and forests. So that's kind of northern Minnesota. But I should point out, I'm going to go back to the other one too. So when you look at the axis, so the number of growing degree days as well, so that occur within a particular week. So if you're going from, I mean, these are increases of 20, 30, growing degree days within that particular week over this amount of time. And so these are potentially meaningful changes within a 15-year time span. Okay. All right. Part two, questions part two of the water temperature. So now we're getting into the new exploratory aspects of this. And so so far you've seen that these changes are not uniformly occurring throughout that annual cycle. They're focused on June and July. So now we're going to ask a couple other questions. Is there an increase in the overall water temperature maximum in the year? Is there also, we want to know if, so we're seeing these increases in June and July, but is there an increase in the overall growing degree days within the year? Meaning, are these increasing trends adding to what we already have, or are there other compensating decreasing trends that kind of make this a net zero gain? So what's happening at the end of the year? Again, this has implications from a fish perspective and from other organisms as well. And then the timing of things. So if things are increasing in June and July, is that because everything's just shifting a little bit earlier? So is the date of occurrence changing? Or is it because there's a change in the rate that things are happening? And so we're going to explore the date of occurrence and the rate of warming in the spring, cooling in the fall. Okay. So start with maximum temperature by ecoregion. So again, the maximum temperatures, if you look at the y-axis there, you have a variety. Again, inverse with latitude, the Canadian Shield Lakes. And some of these lakes are pretty cold, you know, which again, I'm glad we have them. We have this whole spectrum of water temperatures, and all the way up to the plains lakes. But the two movers and shakers are the central two ecoregions in terms of seeing actual increases in the maximum water temperature over this period of time. And both of the central ecoregions. And so that's a big chunk of the state right here that's seeing increases in that maximum water temperature. What about overall growing degree days? This one's kind of interesting because it is increasing, but we've done several iterations of this, and there is a lot of variability in this, and so the trend isn't as clear cut with this one, but in this scenario, it has been increasing a little bit. Okay, now what about maximum temperature, the date of occurrence? So now we have the day of the year on the y-axis there. So in this case, these negative slopes mean that the maximum temperature is occurring earlier in the year than it did 15 years ago on average. Okay, on average, you can see the variability. Like, that's what's kind of neat about these data. Not every year is the same. There's a lot of variability here. So the more years that we can add to this, the better we'll understand the general trends to cut through all of that but this slide does a great job of, yeah, there is a lot of variability there, but on average, it's occurring earlier in the year, because most likely tied to those increases in June and July. Okay, so the date of occurrence for some of these thresholds, I mentioned that 20 degrees Celsius. So as it comes up in the spring, what is that date occurring any earlier in the year, or is it similar? So on the Y, we have day of the year. So a decreasing trend here means that, in fact, it is occurring earlier. The lake is passing that threshold earlier in the year than it otherwise would have. And here's the fall, in the fall, the last 20 degrees Celsius. For two of the southern ecoregions, it's occurring later in the year. So it's a little bit of a delayed fall compared to the northern two ecoregions. I forgot to point out, so way back when I mentioned that Winslow paper and they were showing the monthly differences, did you notice when the greatest changes were occurring? What months was the greatest change? Fall, yes. It was September and October. Is that what we saw? No. So I forgot to point that out, but that is interesting, right? So yeah, I forgot to mention that. I don't know why it just popped in my head now, but it did. So you get bonus points, sir. Okay, now the rate of change. And so here, see, did you bet the over? It's already 40 after. See, the over always wins. Yeah, right. So now, like, now we want to look at the rate of change. So as we're warming in the spring. So you can see from ice out. So like from this period here, and ice out, which roughly occurs on average about 5 degrees Celsius for our lakes. Then there's the period of warming, and it goes all the way up to well over 20, depending on where you are in the state. We want to look at how long it takes to cross different thresholds to go from 5 all the way up, to 10, 15, 20, things like that. So this is our way of looking at that. So we have the day of the year on the y-axis, and then every one of these colors represents one of the different temperature thresholds. So the day on average that we cross that threshold as we're warming in the spring, okay? And what we want to point out here is that if you take this bracket here, so this is for the Canadian Shield Lakes. So this bracket is the same size on both sides of the screen. And so this is the number of days it took in 2000. This is on average. These are, but it used to take 70 days. And over 15 years, that's now compressed to a time of 50 days. So the rate is changing. In this 15-year time span, the rate has increased, which is really interesting. And you can kind of see how those lines kind of converge a little bit. So what'll be interesting as we dive into this a little bit more is paying a little more attention to where exactly some of these things are occurring. If we look at some of the other ecoregions, we notice some similar things where that rate of change is north central hardwoods. That rate of change is seems to be so the rate is increasing, the amount of days is decreasing. And so, but it's not consistent. This one's not as clear cut. Same with the plains. Now, what about the descending limb in the fall? So the rate of cooling. So again, you can see what used to take, and so these are all color coded, and you start at a meaningful number, maybe like a 22 or something like that. And again, that descending limb is taking fewer number of days on average now than it did 15 years ago, collectively in these lakes. There's another this is central hardwoods. And then again, it's not as consistent for the other two. Okay, so to wrap up a little bit, and then we'll go into the biological communities. Well, maybe we will. Looking at the time, we'll see. So far, the changes are not uniform throughout that annual cycle. We saw them kind of focused in June and July. We've seen changes in both the date of occurrence and in the rate of warming and cooling, which is really interesting. And so maybe real quickly, I'll talk about some of the implications for this, because to bring it to kind of a fisheries application, which is really something that we have a lot of interest in. Temperature impacts these systems to a great degree. And I really like this figure here, which is growing degree days plotted against the weight of age 0 largemouth bass. And look at how influential and how predictive that temperature is in dictating the length of fish going into their first winter, right? And it's been shown that survival probability is related to that length. So this is some work that Gretchen has done, and it really illustrates the changes, how water temperature can influence big picture the fish communities. And so it's plotting growing degree days with the probability of a centrarchid versus more of a Persid fishery. And as you increase, it goes to more of a centrarchid based fishery. And we have seen changes in our fish communities within Minnesota. So within the Sentinel Lakes, these are using, gill net surveys, trap net surveys and electrofishing for largemouth bass. And we have seen the fish communities or the species specific changes occurring over time, where there's increases in largemouth bass, different centrarchids, who are warm water fish, also increases in northern pike. And then we're seeing decreases in other fish species, decreases in white sucker, decreases in bullhead populations, things like that. So the fish communities are changing as well over time. And I wanted to point out that this was shown statewide by paper a couple of our colleagues put out there and showed this occurring across Minnesota, which is fantastic. And then the last application I'll point out is to the pelagic cold water fish. So as temperature is changing, the habitat for these fish is changing. And so cold water fish, they live out in the middle of the lake, they relate to, their habitat is dictated by temperature and oxygen and light and that are, available out there. And so as these things change, the fish could potentially be impacted. So it's one of the things that we're looking at. We put a lot of effort toward sampling the pelagic cisco populations. So they don't like warm temperatures, so they like to go down and hide, but they need oxygen to survive. So sometimes they can only go so low, and that anoxic conditions force them up. So in some lakes, they get squeezed and their habitat gets really restricted in the late part of the summer. And so one of the things that we're monitoring is these fish populations in response to their habitat in these lakes. And so we target them using vertical gill nets and hydroacoustic surveys on a subset of Sentinel lakes every year. And it's a fantastic way to monitor the health of these populations. Not only how many are there, but where they are located within the lake is also very important, because again, that's habitat, that's temperature and oxygen related. So that hydroacoustics is just a really neat technology to follow these fish. One of our colleagues, Beth Holbrook, is our resident specialist in this technique. We run the same transects the same time of year every year on these subset of lakes, and so we get really good trend data on these cisco populations. It actually produces you can estimate abundance, and because we get our hands on fish, we can estimate biomass of these fish populations, which is really cool, because normally you don't get biomass of your fish populations. That's hard metric to get. But this graphic just kind of shows one of those transects. All right, with that, I better stop and open it up to questions. But yeah. So hopefully that was a lot to take in. So hopefully that sinks in.
Any questions from in-person audience members?
Yes, sir.
With the program, are you able to monitor duration lights? I saw.
So yeah, we don't specifically, but partners over at EWR in the state climatology office do. So yes, and it's a lot broader than just the Sentinel Lakes. Minnesota has an incredible network, and they have it. They have a map and a really nice website where you can look at particular lakes. Like they'll monitor it in real time as they get calls. They'll post it and things like that. And so they do a really nice job. So it's yeah, state climatology office would be
Throw in one thing with the ice too. Even if it's lakes that nobody's actively watching, like some of our more remote ones, we can get a really
fries.
Yes.
How accessible are these data? Publicly posted somewhere? The second question is how open are you guys or the program more broadly into adding some additional stuff like that, development projects, expanding the scope of what you can have?
Yep, so to your first question, everything that we collect is publicly available. So we're working to get it available through a public facing like data portal. That's in progress. And but right now, the best way is to contact us and we can talk about, okay, what are you looking at? Da, da, da. But everything we collect is available to be used. And then to your second question, so yeah, like we're always interested in if people have an application for these data, you're more than welcome. In terms of adding, so like what normally if we partner with universities on different projects and things like that, for more specific questions, the additional monitoring and collection and things like that is taken up by the graduate students. So the framework, we don't have a lot of capacity to add to what we currently do. But for short periods of time, that's normally, if that's what you were kind of referring to. So then that would normally be the graduate students that would take that up. But we have a lot of ideas of how to add different, like I would love to monitor. We all have ideas of what we could do if we had a few more people or and things like that. And so that being said, like we're also happy to listen to whatever ideas that people have. And so yeah. But yeah, just reach out to us with data requests and things like that too. Yeah.
You're looking at the trend within ecoregions. How do you deal with the spatial non-independence? It's like some are closer to the borders of others, some are closer together. There's like widespread latitude across the glacier lakes, close to the cluster.
We haven't so far. Like, I mean, so that it's representative of that area. Yep. And so you bring up a good point too, but within that region, like, so it's I would guess that the lake features would probably have more to do with the variability than even the latitudinal differences within those regions. The depth, the area, things like that. And that's our next step too, is to look at how some of the different specific characteristics kind of come out in helping to explain some of that variability. So, but you're absolutely right. Like there's going to be a lot of different factors that could help to help explain that. So yeah, absolutely. And that's a great point, because the watersheds vary. Some are really small headwaters type lakes, and some are large and very impactful to these lakes. Absolutely. Yep.
We have a question online here. I'll read this out loud. Thank you. I'm not a fish person. I'm wondering about how fish can respond to these changes. Are fish able to adapt to changing water temperatures over generations? How and how quickly can fish get from lake to lake? Are temps changing faster than fish can deal?
Okay. There's a lot there. Yeah. Yeah. What's interesting to me is, so like you showed this correlation with water temperature and different fisheries responses to water temperature. We've looked at a lot, that's an indirect correlation, right? Looked at a lot of different things in between there to try to explain those in-betweens. And it's hard to get those, mechanisms, those direct impacts are much harder to capture than the water temperature, than the indirect. For how do they get from lake to lake is in Minnesota, a lot of lakes are connected, but then they can also be transported by birds, by clams, there's a lot of different ways. But the question of can they change as fast as the water temperature is one that that we're working on. Like that's a concern. That's a concern. So as an agency, we're looking to prioritize like refuge lakes and things like that. Where can we, because changes are not uniform, and I failed to mention that during the talk, but one of the take homes should be, as a management agency, where do we need to prioritize our efforts? And so it's important for us to understand what we can and cannot do, right? Familiar with like the RAD framework, and working through what are our options as a management agency. So you bring up, it's a great point. But there are, the physiology of these different fish species is, I would guess, fairly finite, and the water temperatures are and the other it's not a take home, but it's that this is 15 years. And so like, this is what happened within this 15 years. And we'll want to continue to monitor what's occurring here. But right. But we are seeing those changes in the fish community structure, and things like that as well. I'm trying to, how quickly can they get from lake to lake? That depends. In some lakes, it's very, it's pretty quick. And I'm from the Alexandria area, and there's a chain of lakes, and it's kind of interesting. We monitor the Cisco population in one of the Sentinel lakes, and it's connected through a chain of lakes. Well, when we sample those Cisco, we're pretty sure we're seeing two distinct growth curves, and we're pretty sure that one growth curve is from fish that are coming in from a connecting lake, and which is really interesting. And then obviously, upriver and downriver has a lot to do with it, and things like that too. But there's other cases where they're isolated, and there's not that mobility to deal with these changes. So yeah, great question though. Yeah.
Thinking about those options that you might have for management, I'm curious about the conversations where perhaps you could be facilitating that evolution, like in Southern program.
So yeah, that's a great question. Those things they're being brought up within forestry, so that assisted migration and things like that. So those are topics that are being discussed at the different agencies right now. And so what role do we play in facilitating that? Yeah, so it's a really interesting time right now with these questions. So yeah, great point though. For fisheries, forestry is already having those conversations, and we're having them, but it's not at the level that they're at. Yeah.
Any other questions? Speaker? Cool. Well, give it up one more time for Casey. And we will have a seminar next Friday at the same time, same place. So hope to see you guys there. Thanks again for presenting.
Thank you. Yeah. You're welcome. 1 o'clock, right on the dot.
Yeah, nice.
Yeah, and I am a resident of college. 0 to 30 line.